Hi, it's Ms. Vitale. This podcast is on the chromosome and is meant for AP biology. Walter Sutton's chromosome theory of inheritance in the early 1900s was linked to Mendel's work from the 1860s. If we compare Mendel's factors with Sutton's chromosomes, we see that Mendel's factors occurred in pairs, as did Sutton's chromosomes. Mendel's factors segregate in gamete production, as does Sutton's chromosomes, and Mendel's factors pairs sort independently, as does the chromosomes. So we look at the history of the chromosome, starting with the fruit fly, Drosophila melanogaster. It's been a favorite use in genetic research since 1909, when Thomas Hunt Morgan began collecting, breeding, and studying them. His lab at Columbia University in New York was called the Fly Room, and it was the center of genetic research for 25 years. The fruit fly has many advantages for genetic research. They breed rapidly, going from egg to adult in 2 weeks. They are very prolific. Each female lays hundreds of eggs. They're small, only about 3 millimeters long, and easy to maintain. They are intricately and precisely built. Dozens of easily identifiable traits make them a great organism for studying genetics. And they only have 4 pairs of chromosomes, 3 pairs of autosomes, and 1 pair of sex chromosomes. Morgan's flies had red eyes. These are considered the wild type, which means they display the most common traits. But at one point, a white-eyed male fly was produced. He crossed it with a red-eyed female, and all of the offspring were red-eyed. He knew that the white eye must be recessive, so he crossed 2 of the F2s and got 3/4 red and 1/4 white in the F2 generation, which is what you would expect, but it turned out that all of the white-eyed flies were males. Half of the F2 males were red, and all of the females were red. Morgan hypothesized that a single gene determines eye color, and that gene is on the X chromosome. The sex chromosomes in flies, just like in humans, consist of an X and/or a Y chromosome. Females have 2 X's, and males have an X and a Y. Many genes present on the X chromosome will be present in 2 copies in a female, but only 1 copy in a male. Males are hemizygous. They only have 1 instead of 2 copies of a gene. If a trait is recessive, a female will usually have a dominant allele on the other X to override the recessive allele. However, a male doesn't have the other allele, so a recessive allele on the X will result in that phenotype. In Morgan's flies, the alleles for eye color would look like this. X with the superscript capital R would be red, and X with the superscript lowercase r would be white. If you crossed a heterozygous female with a red-eyed male, you would get 2 red-eyed females, 1 red-eyed male, and 1 white-eyed male. This is what was responsible for the results that he had. Sex-linked genes are genes that are located on the sex chromosome. The X chromosome contained many more genes than the Y. Each chromosome contains hundreds or thousands of genes. The Drosophila X contains about 1,000 genes. The human X has several thousand. Because chromosomes don't come apart during meiosis, genes near each other on a chromosome tend to be inherited together. These genes are linked genes. This characteristic goes against independent assortment. In fact, Mendel sifted through scores of traits in 34 varieties of pea plants for traits that would confirm his hypothesis. Was he really doing science properly, or was he only including things he could explain and excluding things he couldn't? By eliminating the things he couldn't explain, he was able to come up with principles that have held true, but when you're doing science, you really cannot ignore things that you don't expect or you can't explain. So the principle of independent assortment only holds true for chromosomes, not genes. Morgan's work on sex linkage helped establish that genes are indeed on chromosomes, but where do the genes lie? Morgan crossed two types of flies, normal wings with red eyes, which are the wild type, and short wings, which are also called vestigial wings, with purple eyes. He used PR for purple and PR plus for red, VG for vestigial wings, and VG plus for normal wings. When normal wing red eyes were crossed with short wing purple eyes, all of the offspring had the wild type phenotypes. You see this here in the F1. To see if the genes assorted independently, Morgan did a back cross. He mated the heterozygous F1 flies with purple vestigial flies. You see his results at the bottom. He should have gotten 1/4 red normal, 1/4 red vestigial, 1/4 purple normal, and 1/4 purple vestigial, but in fact, he got 45% red normal, 45% purple vestigial, 5% red vestigial, and 5% purple normal. Almost 90% of the offspring had the same phenotypes as the original parents. Morgan said that the alleles didn't assort independently. The PR plus VG plus stayed together and the PR VG stayed together. He thought that they stayed together because they were on the same chromosome and that the genes assort independently when they are on separate chromosomes, or as we'll see later, far apart on the same chromosome. The tendency of genes to stay together is called genetic linkage. Genetic linkage is responsible for the results of the cross we just did. The 5% and the 5% are the results of crossing over. After crossing over, a new chromosome contains genes from each of the two homologs. These new recombinants stay together for the rest of meiosis. These new chromosomes are called recombinants. Recombinants are offsprings which traits different from the parents. The further apart two genes are on the chromosome, the greater the likelihood that they will cross over. This was discovered by Alfred sturtevant. He was an undergraduate student in Morgan's lab. He realized he could express the distance between two genes on a chromosome by the chance that recombination would occur between them. In other words, genes are more likely to recombine or cross over the further apart they are, and the frequency of crossing over can be correlated to the distance between them. He defined a map unit as the distance between two genes that would produce 1% recombinant gametes, therefore 99% parental gametes. He then constructed a genetic map or linkage map that summarized the distance between the genes. The locus is the position of a gene on a chromosome. Loci is plural. Sex-linked disorders are attributed to the X chromosome. Although the presence of XX or XY determines sex in humans, the genes on these chromosomes don't necessarily have anything to do with sex. Because most disorders are recessive, a female usually has the dominant allele on one X and the disorder is not expressed. A male with one allele on the X doesn't have a matching allele on the Y, and therefore the disorder will be expressed with only the one allele. One example is Duchenne muscular dystrophy. About 1 in 35,000 males in the United States suffer from this. They don't live beyond their teens because their muscle tissue breaks down. It's due to the absence of the muscle protein dystrophin. Colorblindness is when individuals can't perceive certain colors. Red-green colorblindness is the most common. They can't tell the difference between red and green. It affects eye pigments responsible for color vision. People with colorblindness lack one or more types of cone opsin. They usually lack green, sensitive opsin. The retina contains two types of photoreceptors, rods and cones. Rods are for night vision. They pick up light. They're the most sensitive to blue-green light. Cones are for color vision. There's 3 varieties. One that's sensitive to blue, one that's sensitive to yellow, and one that's sensitive to green. Those varieties contain proteins called opsins. Each type of opsin is sensitive to a certain color. Hemophilia, which we've already discussed, is a recessive sex-linked disorder. and the person lacks a clotting protein. Scientists made recombinant DNA by cutting and pasting pieces of genes together that are not found in nature. They're called designer genes. They use bacteria to produce the proteins needed. Instead of giving the protein to the person, gene therapy is now being used. The cells of the person are engineered to produce the protein. Researchers are experimenting with inserting the gene for the clotting factor into cells that can be implanted into the person, or attach the gene to a virus and let the virus infect the person and spread the gene. Right now, the medication for a hemophiliac is about $100,000 a year. There are also chromosomal mutations. These can change the structure of a chromosome, or it can change the number of chromosomes an organism has. Nondisjunction is the failure of homologous chromosomes in anaphase 1 to move apart, or of sister chromatids in anaphase 2 to separate. This results in a gamete having too many or too few chromosomes. After fertilization, the offspring has an abnormal number of chromosomes. This is called aneuploidy. An aneuploid, therefore, is an organism with abnormal chromosome numbers, and a euploid is an organism with the correct chromosome number. Trisomic means there is one extra chromosome, so the cell is 2n plus 1, diploid plus 1. Monosomic is a missing chromosome, so the cell is 2n minus 1. Nondisjunction can also occur during mitosis. The effect will depend on the stage of development. Polyploidy is when chromosomes don't separate normally during mitosis or meiosis. It is the nondisjunction of all chromosomes. It results in multiples of chromosomes, so you can get a 3n, a 4n, a 5n. This is commonly found in plants, and it's actually chemically induced by plant breeders to produce larger fruits and flowers. It is always fatal in animals. Deletion is when a piece of a chromosome breaks off, resulting in the loss of genes. Duplication or addition is when a piece of a chromosome breaks off and attaches to a homologous chromosome. Translocation is the transfer of a part of a chromosome to a non-homologous chromosome. An inversion is when a piece of a chromosome is rotated, which reverses the order of the genes in that segment. Chromosomal disorders are any of the above mutations that can result in disorders. Often, additional or missing chromosomes result in the death of the embryo in humans. For certain aneuploid conditions in which the fetus does survive, the individual is said to have a syndrome, which is a set of symptoms. Down syndrome is trisomy 21, when there is an extra 21st chromosome. These individuals have mental retardation. They have abnormal development of the face, heart, and other body parts. About 1 out of every 700 Americans are affected. They tend to be sexually underdeveloped and sterile, and the risk rises considerably in older women to give birth to a child with Down syndrome. Women over 35 are advised to have fetal testing to see if this is the case. Klinefelter syndrome is an extra X in males, so they are XXY. About 1 in every 2,000 births is Klinefelter syndrome. The males have underdeveloped testicles and are sterile. They may develop feminine characteristics like breasts, but hormone therapy during puberty usually resolves these issues. Turner syndrome is nondisjunction of sex chromosomes. They only have one X present. This is a female who doesn't develop sexually. Her sex organs don't develop, and therefore she is sterile. She usually doesn't reach full height. Normally the intelligence is average, and it is the only known viable monosomy in humans. These girls can also receive hormone therapy and develop more normally. We sometimes see normal chromosome numbers, but with structural abnormalities. Cri-du-chat is French for cry of the cat. These children are born with the deletion in chromosome 5. They tend to be mentally retarded. They have facial abnormalities. They don't live a full lifespan. And the name of the disorder is because their cries sound like cats. CML is a cancer that affects the cells that make white blood cells. In the cancer cells of people with CML, a translocation has occurred. A portion of chromosome 22 has switched with a portion of chromosome 9. Imprinting sometimes occurs in mammals. The effect of a specific allele is different depending on whether it comes from the mother or the father. This is not necessarily on the X chromosome, so it's not the same as sex linkage. An example is when there's a deletion of a particular segment of chromosome 15. If you inherit that chromosome from your father, you have Prader-Willi syndrome. These children are mentally retarded, obese, they're short, and they have unusually small hands and feet. If you inherit the defective chromosome 15 from your mother, you have Angelman syndrome. These children have uncontrollable laughter, jerky movements, and other motor and mental symptoms. Genomic imprinting is when a gene on one chromosome is repressed while its allele on the homologous chromosome is expressed. The same allele will act differently in an individual depending on whether it was inherited from the mother or father. An allele from an ovum may be different than the same allele from a sperm. It seems to be important that an animal get an imprinted copy that inactivates the other copy. There is an abnormal X chromosome in Fragile X. It is a syndrome that is inherited from the mother rather than from the father, and it causes mental retardation. Extranuclear genes is DNA that can be found in the mitochondria and the chloroplasts. Mitochondria and chloroplasts reproduce and pass their DNA to their daughter organelles. DNA in the mitochondria and chloroplasts comes from the female parent, from the mother, because you only get cytoplasm from an ovum. Sperm do not have any cytoplasm. The products of this DNA genes are the proteins in the electron transport chain and in ATP synthase. If the protein is defective, ATP production is affected. Mitochondrial myopathy causes muscle deterioration and weakness. We can also use mitochondrial DNA to trace lineage.